Hello everyone. Today we'll be going to discuss about a very important topic, that's genetic linkage. Earlier as we have gone through that after the onset of Mendel's law of inheritance, things have been changed a lot. Since the Mendel's law was completely based on the alleles and their characteristics, later on, after the death of Mendel in the initiation of the 20th century, that is 1900, Sutton and Boveri, they were the two renowned scientists and personality of that time. They proposed the chromosomal theory of inheritance, which was in about year 1902 and 1903. As per the theory says, it is well established that many genes are located in each chromosome in a linear fashion. As you know that lot of genes are present within a segment of DNA, we can say that whole DNA is not a gene, just a specific part of gene of that DNA is called a gene, and that codes for some or a specific RNA which further leads to production of proteins. All genes located in same chromosome would move to same pole during cell division. Yes, definitely during a cell division process, all the genes are just passed to the next generation, and in the meiosis state they move to the poles. As a consequence, such genes will fail to show independent segregation and would tend to be inherited together. But as since the genes are moved to the ending poles of the cell, so there are conditions that both the genes or a segment of genes, you can say 10 genes, 20 genes, 30 genes, they should be inherited similar in a similar pattern. But that doesn't rarely happens. So coming to the point, what is a genetic linkage? Genetic linkage is the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of asexual reproduction. Or you can say the tendency of genes to remain together in their original combination during inheritance is called linkage. As the word linkage, link suggests when 2 or 3 genes or 10 genes or 30 genes, if they are linked to one another or they are present randomly one after one another, there are a lot of chances that they will be inherited in the same pattern, like gene A, B, C. If they are present in a sequence, there are a lot of chances they will be expressed and transferred to the next generation the same pattern. We can also say that the two genes that are physically near to one another are unlikely to be separated onto different chromatids during chromosomal crossing overs, and are therefore said to be more linked than genes that are far apart. Yes, if two genes are located on nearby within a chromosome, there are a lot of chances for them to be transferred in a same pattern. Also, in other words, the nearer two genes are on a chromosome, the lower the chances of recombination between them. Vice versa, we can say more that if two genes are linked to one another, there are rare chances then of recombination in that case. Why and what is the need of a genetic linkage? Because in our organism, genes are far more in number than the chromosomes, which implies that each chromosome contains many loci. Definitely, just talking about a genome size, that is 3034.83 megabase pairs per haploid genome, or you can say 649.66 MB total diploidy, and the number of chromosomes in humans is 23, as you can say 46 pairs. So there is indeed a need of a recombination and a linkage with that. So coming to the point, when and who discovered the linkage phenomena? Shortly after the Mendel's work, as I said previously, expectations to these rules were found. In 1906, William Bateson and the Punnett discovered the phenomena of linkage in sweet pea. As earlier in the case of Gregor Mendel, we were postulating that the genes are transferred independently, as said by the law of independent assortment, as per the Mendel's theories. But when the scientist named Bateson came, he postulated another theory that there is a variation. There occurs a chances that the two genes can be transferred, or two alleles can be transferred togetherly, dependent on one another. So the question comes in mind, how it was discovered. The researchers crossed variety of sweet peas that differed in two traits: flower color, pollen length. Plants with red flower and long pollen grains were crossed to the plants with white flowers and short pollen grains. All the F1 plants had red flowers and long pollen grains, indicating that the alleles for these two phenotypes were dominant. When the F1 plants were self-fertilized, Bateson and Punnett observed a peculiar distribution of phenotypes among the offsprings. Instead of 9:3:1 ratio expected for two independent assorting genes, they obtained a ratio 24:3:1.1, 1:1:7. That is, it's clear that the ratio was not as expected as per the Mendel's results. There were chances of linked genes among that. So now a question comes in our mind, can linked genes be separated? Yes, obviously there are chances linked genes can be separated with the phenomena of recombination. As telling it in briefly, recombination is a process in which two linked and we can say unlinked genes recombine themselves and something new is formed. Recombination, explaining it in a deep manner, recombination is a process through which genes present on same chromosomes can be separated and new combination of genes could be formed. Recombination is caused by a physical exchange between the pair of homologous chromosomes early in the prophase of the first meiotic division after the chromosomes have duplicated. That's a very essential phenomena which occurs in the periodic phase of meiosis. As we can say, recombination process which genes present in same chromosomes can be separated, like a specific chromosome if it's present. So we can say from a same chromosomes genes we can do the mixing phenomena so that 2-3 genes are transferred and we can say that they are leaving the phenomena of linkage. So at any point along a chromosome, the process of exchange involves only 2 or 4 chromatids in a meiotic tetrad. Later in prophase 1, crossovers become visible as a chiasmata. Who provided the evidence? Initially in 1931, Harriet Crichton and Barbara McClintock obtained the evidence of this specific phenomena of recombination. So now what is the recombination frequency? Comes a question in our mind. The frequency of the recombination progeny produced by the heterozygous F1 plant is called the recombination frequency. As we can say, a heterozygous F1 plant is a plant which is having two different types of alleles, not specific. For example, capital T and small t. Genes that are tightly linked seldom recombine, that is their recombination frequency is low. So we can say if two genes are linked very tightly, we can say that they have a good linkage. So you can say indirectly proportional. Recombination is indirectly proportional to a linkage phenomena. So that's an important part. What are the factors affecting recombination frequency? First, distance between the genes. Second, sex. Heterogametic sex shows relatively low recombination. Age. A lot of recombination factors depends on age, temperature, nutrition, chemicals, radiations, chromosomal abbreviations, distance from the centromere. All these factors play a crucial role in the recombination frequency. Now there are the classification of types of linkage. One is a complete linkage. When one parental characters combinations are recovered in test cross progeny, it is called a complete linkage. Its common example is the male Drosophila. Incomplete linkage occurs when recombination types are also recovered with parental types in the test progeny. It is called a incomplete linkage. So there are lots of examples, and one is the sex linkage in genes present which occurs in the sex chromosomes. So what is a linkage group? All genes are linked together to form a linkage group. The commonly Drosophila has 5 linkage group, and depending on species to species and organism to organism, there are variability in lots of linkages. So what is a chromosome map? How it is created? It is a simplified version of getting the knowledge of what is the level of recombination and what is the level of the linkage among two genes. So friends, briefing about it, It is a very important factor, and linkage and recombination overall plays a crucial role in transferring of genes, and also they are a very important part of maintaining the similarity passed to the next generation, and also they play a crucial role in the evolution process. Thank you. And for the next lecture, we'll be discussing guys another interesting topic. Till then, thanks.